Okay, thank you very much for the introduction. Thank you for being here. So the game for me is to try to tell you how much over the last 7 years we've made efforts to bridge the gap between Python and brain time series and using this package that we call MNE. So just to give you the objectives for this talk, I don't know how many of you are actually familiar with what I call neural time series or brainwaves. So I'll just take a few minutes to show you what types of signals we're playing with and trying to make sense of, and then I'll show you how MNE can work with these time series. What can you roughly do with it in very few slides, and then I'll have prepared some Jupyter slides that you can download at this link at the bottom. That will you can just run them. It will fetch the data online from Amazon, and you can run every cell on your system. Play with it, break it, do whatever you want with it. Okay, so let's start really quickly, but what are neural time series? So roughly speaking, when you have your neurons that are active, you have ions, so electric charges that move somewhere on the dendrites or on the axons and different places in your brain. And when you have electric charges that move, they create electromagnetic fields and that you can, in the end, measure. So active neurons produce these difference of potentials, magnetic changes. This is what we're going to be recording. The nice things about this is that these can have very high sampling rates. So we typically work with time series which are the order of 1 kilohertz or multiple kilohertz, which is very high resolution compared to other ways of imaging the brain. So if you're wondering how we measure these types of things, this is a very old picture that you see at the bottom of the historical setup of EEG recordings. And where basically you stick electrodes on the skin of the people and you measure differences of potential, and that gives you time series of potential in volts or microvolts on the order of a few microvolts. You can also use another technique called MEG or magnetoencephalography, which measures the magnetic field which is produced by the brain, and then you can record with very fancy sensors that we call squids, which live in a very low temperature medium, and that records things on the order of a few femtotesla. So very, very tiny fields that also produce these types of time series. Now, if you want to go invasive and you have like patients, or you can remove a chunk of the skull and you can put electrode grids on it. That's called epochs or electrocorticography. And each of these grid points, you measure also the local potential that also gives you time series. Or if you're in Europe and you don't use grids because grids are mostly used in the US, you stick electrodes inside the brain. So this is what you see on this slide. You have these very long electrodes that you stick into the head of people. So don't worry, it's not very harmful. You don't really feel anything there. And you end up recording from a few dozens of positions inside the brain the local potential. So enough of a story about these time series. The take-home message of all this is MNE is a Python package, a pure Python package relying on the entire SciPy stack to process, visualize, and work with these types of high sampling rate time series. So if you go to the website of MNE, you'll see a lot of information. And again, so we are pure Python, rely on NumPy, SciPy, Matlablib. We use Mayavi for 3D rendering, and also things like JobLib for parallel computation. And I think I forget stuff. But roughly speaking, the hard dependencies are NumPy, SciPy, and Matlablib. Okay, so this is a screenshot of the documentation of MNE, going from what the containers are. I will illustrate this in a minute with examples, the types of I/O we can do, the types of preprocessing we can do, how do we visualize, and how do we go up into the source space, which is where things happen in the brain using these types of time series. And also, I will do a quick demo how we can use scikit-learn to figure out where the signal is coming from and if there's actually signal and predictive power in the types of brainwave we record. So let's import MNE and see what is there. So I think I will show you real quick the data I'm going to play with. So this is a classical cognitive paradigm where you put the person in an MEG system here, and you present them either a phase or a scrambled phase. Okay, the typical two-condition contrast, and you record this. You do this multiple times, and you want to figure out where in the brain is processed the information about phase processing. So let's do now the live demo so you see a bit more how we do this with MNE. So if you want to download the materials, just bit.ly. I think the link is wrong. It's bit.ly/MNE_SciPy17. All right, so I've got these live slides that I won't play with you because I've got too little time. If anything breaks, I will be in big trouble. So I'll just show you the rendered slides. So let's import MNE. So we just have a logging system. So here I just only show warnings so you don't see all the logs, otherwise your screen will be completely filled with info messages. So MNE gives access to a lot of public data. I think it's one of the strengths of MNE is that you can just download any example on a website, either the notebook version or the plain Python version. It will fetch the data if you don't have it on your system, and then you can execute the script. Basically, it means that the barrier of entry for other people to use MNE is very low, hopefully. So here I'm going to use a data that contains this, the data for one subject following this paradigm. Is it a phase or a scramble phase? And here, what you see here roughly is just the file name, and here I'm just going to read it with CTF because it's the data coming from a CTF system. In the end, I've got a raw object, which is the basic container that we use for raw data. And you see that roughly the size of the data in memory is a little less than a gigabyte. And so what else can I tell you? We have 340 sensors, and this is the number of time samples over the length of the recording. So let's do very simple pre-processing. You can do much more, but typical pre-processing is done via methods on the raw data container. And here I'm just going to do a filtering between 1 Hz and 40 Hz using a certain filter design, and eventually setting a number of jobs that I can use in parallel using under the hood joblib parallel. If you're wondering why we don't use plain NumPy arrays, it's because we have a bunch of metadata to associate to each of these data. So roughly, we call this measurement info. So every data container has an attribute, which is a measurement info that here is just basically a dictionary, so a pure Python dict with a nice rep that allows you to print these in a readable way. So this is all the metadata that we typically store on all of the objects in order to track a bit what was done to this, and what is the channel types, and what are the sensor positions, and a lot of these types of important information for further down processing. So if we can look at the data using Matlablib, so this is a kind of fancy Matlablib GUI that you could also run in the notebook with the notebook backends that allows you to interact with the sensors in time, and also vertically by looking at more sensors with also a lot of shortcuts. So I didn't realize how many creative things you could do with just Matlablib and in terms of advanced GUIs. Typical things that we do, we look at power spectrum density. So this is just looking at the power spectral density of all of our raw container. And the first time that you see here is what we call a head shape, a head cast. And all of these dots, colored dots, is where the sensor is coming from. So we use typically color to say where things happen on the helmet or on the head. Epochs and events. So when you do these types of experimental paradigm, you present a phase or a scramble phase. It means that at a certain instant in your recording, you have an onset of a stimulus. This is what we call events, and an epoch is just a chunk of data, a tiny interval of data around an event that's basically going to capture what's happening just before and what's happening just after the phase presentation or the scramble phase presentation, that will contain what the brain actually produce in terms of signals. So typically in the data, we have what we call a steam channel that records when things happen during the paradigm. And here I'm just going to do MNE.find event of my row object, and I'm going to get an events array, which is just a NumPy array that says at what sample there's something happen, and here what is the types of event that happened. We can look at the design. So this is typical design. This is seconds in the x-axis, and here what is green is a presentation of a scramble phase, and blue is when a phase was actually presented. So you see that it's done multiple times, roughly 80 times in this experiment, so something like 160 image presentations. So we use this dictionary to specify what, like in human readable way, what is the condition and the associated number in the trigger channel. And now from this paradigm, we can extract the chunks of data that are in it and extract what we call epochs. So here, taking 100 milliseconds before, half a second after, selecting the channels that we're interested in, and then we create this new data container, which is all the single presentation, all the chunk of data that correspond to a single presentation. And in the end, I've got my epochs, which are 167. This is the size of the data, so you see it's much smaller, and the number of time it corresponds to a phase or a scramble phase. Let's try to open the box and go to low-level NumPy. So I can get the data from these epochs, and I've got all my epochs presentation. This is 305 channels, and this is 145 time points. So epochs.times give me an array of time instance in seconds, so I can convert to milliseconds. This is the data, and this is typically showing you, for the first epochs, the data that we collect. Okay, this is just exposing an NumPy array from our epochs data container. If you want to denoise things, the typical way to do this, you have all these presentation of an image, you average all these repetitions that basically kills the noise and keeps the evoked response of the brain. And this is what you get. So you see just by computing mean on the axis 0, which is the epoch axis, then you see that at 0, there's nothing much happening before, and suddenly there's a kind of a brainwave occurring, and this is what people call an event-related field or an evoked response. And you see that the peak of the response is roughly starts before 100 milliseconds, lasts 200 milliseconds, but there's actually still some stuff happening later, but at much slower amplitudes. Basically, MNE offers you a possibility to do all this using all the metadata in a very convenient way. So here I've got my epochs, I can just average them, and I get something that I call an evoked data container that I can just plot using spatial colors, and you see that every line now tells me where the sensors are. Okay, so you see that there are different components appearing in data. I can also plot things that are called topographies, which is where on the helmet or where on the sensors do we have actually strong signals. So you see this over time by specifying at what time instant you want to plot things. You know that there is a strong effect appearing after 180 milliseconds. What else can you do? You can also plot them jointly. So you can all of this is done with plain Matlablib. And what you see for every instance, the corresponding topography. So this is kind of very like summary image that people expect to see from these types of experiments. You can do this interactively. So this is a tiny attempt to use interact using IP widgets. So you can move over time and look at the topography and how it evolves at different time instance. So I was saying that at around 180, you have a strong field. So this is what you see, hopefully, here. Okay. So you see basically the evolution of the signal over time using interactive widgets. You can use typical things that people start like doing on these types of multivariate time series, which is independent component analysis. So this is under calling some methods from scikit-learn, and to show you what are the latent sources in the data. So here you call this on the containers that we have. You have an ICA object and you just fit it. So if for some of you that know the scikit-learn API, this is very inspired by the scikit-learn API. You fit your models and then you can transform, you can and do many types of things with it. So here, this is a bit more than a scikit-learn estimator. You can just call it plot functions, that you can plot the components, and you can plot the corresponding sources. And it turns out that these components, you will have to believe me, it corresponds to the heartbeats of the subject. then typically you would remove it from the data if you don't want to have corruption from the heart of the people. Where is things coming up in the brain? This is kind of the actual question that interests me is, can we use these external recordings to figure out where things happen in the brain? So I've done something which is very, very standard and very classical in our community, which is try to take one time point, which is a time point where it's actually something happening in the data. So here what you see, this is the nose, this is the ears of the people, so you roughly have signal that come from the back of the head. So we can use the anatomy of the subject. This is a T1MR image that we segment automatically using FreeSurfer. And then we use all these FreeSurfer outputs to build a forward model that says how a source in the brain relates to the sensors. So all this is all the BEM computation, the boundary element methods, all of this is computed in Python using efficient NumPy tricks. And this allows us to actually invert the system to know where things happen in the brain. So here, this is DeltaPlot using MAIAVI. MAIAVI is always a bit of a nightmare for me, especially notebooks and various systems, but still I rely on this for doing these types of plot where you see the head and the sensors. And then we have this function called FitDipole, and you can just see this as a way to call SciPy optimize to know where to look for the signal and optimize the position and orientation of the source to explain the data. All this, you need to know the geometry of the sensors, you need to know many things, and MNE basically encapsulates all this. And in the end, you can do these types of plots. This is, again, pure Matlablib using the 3D capabilities of Matlablib, where you see on the MR slices where the dipole is, and hopefully, this seems to be correct because it's actually coming from the back of the head. Okay, so we use all these raw data, we compute the epochs, we average them, and then we use the actual head model coming from the MR of individual subjects to know where things happen in the brain. We can also do machine learning to classify if the person saw a face or a scrambled face. I don't have time to go into the details, but basically the only thing that bothers for me right now is with just 10 lines of code using scikit-learn and MNE, you can say that with an accuracy of 93%, you can classify if the subject saw a face or a scrambled face. And you can see this, you can do this at every time point, and for example, see that when in the data is the effect of interest? When can you classify, from what time segment after the stimulation can you classify if the subject saw a face or a scrambled face? So that's it for my demos. I'm going to go back to my slides and try to conclude in 2 minutes. We can also do these types of more fancy plots using Matlablib. So this is just source reconstruction with a millisecond timescale. And we also have a way to generate static web pages that we use for quality control and what we call the web reports. So we save this static HTML file that embeds everything, the JavaScript and the PNG or the SVG that we produce, and basically we can hand this to people, and my grad students give this to me, and I can check the different steps of their pipeline. So MNE is a very distributed development effort. Many people have contributed to this from very different labs, open science people, and the idea was really to share best practices across different labs. And over the last 7 years, you don't imagine the number of tricks that I learned from people in Germany, in the US, in Canada, that shared all their tricks that I had come up with over the last 15 years. And it was just sitting in a hard drive somewhere. So this is all the people that contributed to MNE over the last 7 years. Many people. Hopefully more soon. And also, to conclude, this is an effort that was made possible by a lot of people in this conference, some of you in this room, and an entire ecosystem of open source packages and GitHub integration services that allow us to scale the development to many people. So I think that's it. Thank you very much for that attention.
We have time for questions.
You can also comment and critics. It's fine. I'm French. I'm used to it. Yeah.
So many attribute analysis in Python today happens in pandas. You chose not to go that route. Can you speak about why you made that choice? Or
That's a very good question. I guess when we started MNE, pandas was not as mature, and the time series module was not there. Probably that could be reconsidered, or that could have been reconsidered over time. It was just a big efforts to do this. Yeah, but that's true. The pandas. I mean, right now we have a soft dependency of pandas, so you can take all the data containers and you can export as data frames using a time index. But we don't internally use pandas for all the numerics. Yes?
Question about your classifier. How many features did you use? And is your classifier patient specific, or is it patient?
So the classifier is just working here on individual subjects. So I take a chunk of data from one subject and I predict on the same subject on new epochs. Now, if you wanted to like predictions on left out subjects, MNE doesn't really expose the right tools to do this, but there's a package written by another French guy called Alexandre. It's called Pyreman, which is a very clever way of generalizing your classifier to a new data sets which have been kind of finely transformed due to different head geometries. Yes?
Inside the library, do you guys have a lot of stuff for like cleaning EEG signals, stuff like that?
Yeah. So typically we use ICA. You can manually annotate bad segments using the Matlablib viewers that we have. So you can select like time intervals. So you can eyeball the data to do the cleaning, or you can rely on things like ICA or just peak-to-peak differences. If the peak-to-peak is too big or the variance is too big, then you remove the segments. And you can basically annotate the data, say this is a bad segment, and then all these bad segments end up being ignored, for example, from epoching for power spectrum density computation. Yep.
I was just curious how you handle testing. Like if you have like a workflow and you have some analysis that depends on previous results you got from data, how do you do that like on Travis?
So Travis does all the unit testing, and including tests on some of the data that we ship. So actually for the big data, we use Circle CI that has a much bigger capacity for caching the data sets. I mean, at least at that time, it was clearly the better way to do it. So when we regularly, I mean, every time we push on a PR or on master, we can rerun an entire analysis coming from using the 2 gigabytes of raw data, epoching. And so this is really for the testing on the website and the documentation. Now for the internal, like the science that we do with it, I mean, I tend to rely on scripts and say that everything in the end is very CPU time. It's cheap. Nothing takes too much time. So if things should rerun with your scripts in a few hours, and yeah, that's my approach.
Last question?
Yeah.
So one comment, the vendor support is impressive in terms of being able to read in things and do common processing for different vendors. But one question is, do you have any real-time interfaces to any of the acquisition systems, or is it strictly a post-processing?
So there's actually a real-time module in MNE that was contributed by a GSOC student many years ago. It was plugged with the field trip buffer at that time. There's also now some people in Finland that have written a real-time connector with the acquisition system of one MEG system. And there's some people that are unfortunately not here that work with OpenBCI in Stanford and Berkeley and try to hack something on top using MNE and glue it to a cheap EEG headset.
Let's thank the speaker again.
Thank you very much.